Hi, welcome to the Bioinformatics chat. Today we will be talking about plasmids, and joining me today are Anita Schürch. Hi, Anita.
Hi.
And Sergio Arredondo. Hi, Sergio.
Hi, Roman.
Let's start with a brief primer on plasmids and maybe on bacterial genetics in general.
We are interested in plasmids because, plasmids in general, there are sequences, autonomous sequences in bacterial cells. So we have a chromosome and often extra chromosomal elements, plasmids, which can replicate independently.
So bacteria, they, like eukaryotes, they have chromosomes, right? But usually just a single chromosome?
So you have a single cell indeed, and there's a single chromosome inside it, most often a single chromosome, and which is basically like a single string of DNA. But next to this single chromosome, we often have also other DNA elements, which are also circular, like the chromosome, but most often much smaller than a chromosome. And these pieces of DNA, plasmids, they can replicate independently of the chromosome. So they are like small selfish elements within the bacterial genome.
And when you say they replicate separately, what does it mean? Do they have their own sort of replication cycle, so they replicate more often than the main chromosome?
They have their own replication machinery, indeed. And they can be in a much larger copy number than the chromosome, but that's not always the case. Especially if we have bacterial plasmids which carry antibiotic resistance genes, these do often replicate at a similar rate as the chromosome. So they have also a single copy number. And the resistance genes, the antibiotic resistance gene, is also one of the reasons why we are so interested in plasmids. So we work often with commensal bacteria, enterococci, which live in the intestines of everyone. So every person carries them, and they don't do much harm, but they can also trigger infection, especially in hospitalized patients. And the bad thing is that they are also often associated with antibiotic resistance. So the antibiotics that are used to treat these infections, they become resistant too. And these resistance determinants, these genes that confer the resistance, can be located on plasmids.
Mm-hmm. And so what differentiates plasmids from chromosomes? Well, first of all, plasmids are smaller, right? But also they can be more freely passed from one organism to another, from one bacterium to another, right? And that's how they spread this resistance.
Yeah, that can be the case. So these plasmids can be transferred between bacteria. It's what we call horizontal transfer, as opposed to vertical transfer, where a mother cell gives the genes to the daughter cells. So that would be one mode of transfer, and that's mostly when genes are located on the chromosome. But if they're on the plasmid, they can be horizontally transferred and also very rapidly transferred between different bacteria.
Right, and knowing whether a gene resides on a chromosome versus a plasmid, it may be pretty important, right? So you analyze some data from vancomycin-resistant enterococci. Can you talk about how that problem arose and what you found there?
Okay, so we started to work with enterococci, and enterococci are bacteria that each of us carry. It's usually in our gut and doesn't pose any problems to healthy people. But it can lead to infection, and then mostly in hospitalized patients. And it's also very prone to picking up antibiotic resistance. So you would treat enterococci, for example, with vancomycin, which is a very strong antibiotic, but they also tend to get resistant against vancomycin. And we see this vancomycin resistance enterococci often in hospitals all over the world and also in the Netherlands. So what we did was to collect vancomycin-resistant enterococci from Dutch hospitals in order to map the transmission of this vancomycin resistance. We were interested if the transfer of this resistant determinant would be horizontal, thus with plasmids, and between bacteria, or vertical, just from a mother cell to a daughter cell.
Right. So in that study, did you know in advance what gene you were looking for? Like, was it easy to identify the gene itself in, let's say, the sequencing data?
So usually it's quite easy to identify the vancomycin resistant gene. And there are like different variations of that resistant gene. Can be, for example, VAN-A, VAN-V, VAN-D. So usually that's not a problem. So we can easily identify what is the gene variation which is present in a bacterial isolate. It's quite difficult to know is like whether that particular gene is present into the chromosome of the bacterial cell or if it maybe is located in a plasmid sequence.
Do I understand correctly that the same gene might reside, like in some organisms, it might reside on the chromosome, in others on the plasmid? Like, is there, for example, recombination happening between the plasmids and the chromosome?
Yes, that could be the case. So sometimes we find, for example, the vancomycin-resistant gene A, we can find it in the chromosome, but usually it's more located in the plasmids. But it can also be the case because they are present into transposon sequence, so that transposon can also jump into the chromosome. So it differs a lot between the bacterial isolate that you're studying. So what we can see is like there's usually a preference for a particular resistant gene to be located into the chromosome or to the plasmid, but you can also find it in the other way around.
And so you wanted to figure out, right, whether a given, let's say, sequence, right, that comes from the sequencer, whether it comes from the plasmid versus the chromosome. And that sounds like a very hard problem, because how can you possibly distinguish these, right? I understand like perhaps the only difference between them is that plasmid is a smaller chromosome, right? So how is it possible to algorithmically distinguish sequences from plasmids versus chromosomes?
So we actually asked the same question ourselves, and this is why we developed a benchmarking of different tools during the first part of our research in order to see what kind of features we could use in order to predict plasmid sequence. So there are like 3 main features that you can use, and they can give you hints whether a sequence comes from a plasmid or comes from a chromosome. The first is, for example, the presence of replicon genes. That's what Anita was talking about before. So plasmids, they carry their own replication machinery. So if in that part of the sequence you also find replicon genes from plasmids, that can indicate whether that particular sequence comes from a plasmid.
And that gene would code for some protein, like for polymerase or something like that?
Yes, exactly. It's quite similar to the DNA A that you can find in the chromosome. So it's usually a replicase or relaxase. So it's part of the replication machinery of the plasmid. Yes.
Mm-hmm.
As I was saying, there's also 2 other features that you can use in order to distinguish sequence that come from plasmids or from the chromosome. One is, for example, the coverage that you encounter into those contexts. So coverage can be translated into the number of reads covering a particular nucleotide in your context. And that's also related to what you were discussing before. So because plasmids can be in a different copy number than the chromosome, you will expect that the coverage of the context belonging to the plasmids is going to differ, are going to differ respect to the chromosome. So for example, if a plasmid is in a copy number of 5, you will expect that the coverage of context coming from that plasmid is 5 times higher than the coverage from the chromosome. So usually using that feature as well, you can really distinguish plasmids from chromosomes. There's only one limitation behind, which is plasmids that they have a very similar copy number to the chromosome, because then the coverage is going to be very similar to contigs coming from the chromosome.
And you said something that I think we should elaborate on. So you said contigs, right? Which implies that before you do this analysis, you sort of try to assemble. So you have your short sequencing reads, and you try to assemble them the same way we assemble genomes. This is a genome in a way. And you work with these longer sequences because that allows you to find, for example, whether several genes are on the same DNA strand.
Yes, that's exactly correct. So what we usually do is whole genome sequencing of our isolates, and then we usually use Illumina read sequencing. So we have short reads that can differ between 150 base pairs to 250 base pairs, depending on the technology that we are using. But then we try a de novo assembly using, for example, a Spades that does the Bryan graph assembly. And then in that way, what we try to do, as you have said, we try to elongate those reads into longer pieces. That's what we call contigs, because in that way we can have more information in order to predict whether something comes from a plasmid or from a chromosome.
And you mentioned the features of the sequence of the contig that you use to predict the plasmid DNA. So one is the presence of these replication genes. The other one is the coverage. And what's the third one?
It's actually the K-mer composition. So there are some tools. For example, one that we have developed, it's called ML Plasmids, and that's based on 5 Mears frequencies. So the pentamers frequencies that you can find in plasmids and chromosome can differ. So what we actually did was to train different machine learning models using datasets in which we knew whether a contig was from a chromosome or from a plasmid. And then we tried to train those models in order to predict whether a contig was a plasmid or a chromosome. So actually, if you think about it from a machine learning point of view, it's a classification problem in which you have 2 classes, whether something is a plasmid or is a chromosome. And then from each prediction, you have a probability that the contig can belong to the plasmid class or to the chromosome class.
That's very interesting. And why, what made you make this conjecture that the K-mer composition would indicate whether a gene comes from a plasmid versus chromosome? Like, it sounds like you would just be learning the identity of those plasmids. So, of course, if you sort of remembered, right, a big data set of like many chromosomes, many plasmids, then you might be able to classify just based on the similarity. Or do you think that the K-mer composition reveals something about the actual biology of these sequences?
Yes, that's a really good question. I think it's quite difficult to know what actually the models are using in order to classify something as plasmid or chromosome class. I think our feeling is like because of the chromosomes are shared between all the bacterial isolates that we are using, so they are much more conserved. So usually the composition of those chromosomes and those contigs belonging to the chromosome is much more stable. And rather than the plasmids, they can come from different backgrounds. As Anita said, those plasmids, for example, can be transferred from different bacterial species. So actually the composition of those plasmids is going to differ from the chromosome.
And what sort of predictive models have you tried to classify based on the K-mer spectra?
So we actually trained 5 different models. So we tried with logistic regression. Then we also went for decision trees, random forest, support vector machines, and just a simple Bayesian classifier. And then we compared them in our test set, and based on the raw curves, we decided to stick for support vector machines, also because the probabilities of being plasmid or to the chromosome class were very high, which indicated that the prediction was actually we could trust in the prediction from the model.
And you said you're using Illumina to sequence your isolates. Have you considered using long reads? Would long reads address some of the issues with short reads and like having to assemble them?
Long reads can definitely solve the assembly problem and can help you to get completely resolved genomes, thus chromosomes and plasmids. So yes, that helps a lot, and we also do use them, especially to train the ML plasmids classifier. The problem is that at the moment we cannot long read sequence as many sequences or as many genomes as we would like. So if we have thousands and thousands of samples to be sequenced, I think the decision at the moment will still go for short read sequences, just cost-wise. So in order to get complete genomes, what we do with a selected number is that we combine these short read sequence genomes with long reads and do a hybrid assembly. And so that's one point, the amount of sequences we want to generate, the amount of genomes we want to sequence. And the other point is that in the databases, there are still a lot of data from short read sequence genomes, from which we also would like to know if there are plasmids in there and what kind of genes they carry.
That makes a lot of sense. And in this specific study, I understand you're interested in a specific bacterial species. So I guess you try to culture them and then sequence just these enterococci. Do methods work equally well when this is like a metagenomic sample of many different, like a mixture of many different bacterial species?
So actually, for example, if you're working with a different bacterial species, what we have seen in G+, for example, the new tool that we are presenting is like the initial classification, whether something is plasmid or chromosome, is very important. So for example, if you are using a classifier that has been trained for a general purpose, so for example, for using in the test sets or the training sets context from very different bacterial species, then usually the classification is not that good, and that can cause problems in the later binning of the plasmids. So, and of course, at this point we are only working with single isolates, so we're not working with metagenomic sequence, because as you have said, metagenomic sequence is like a much challenging problem, because you have different bacterial species co-occurring in your assembly, and you also face other problems that you don't face in when you are working with single genomes.
Now, you mentioned that you use spades for your assembly, and there is also metaspades, which I guess is not applicable because what you just said, that you're not working with metagenomic samples, right? But there's also something called plasmid spades. Can you talk about that and how it's different from like the normal spades?
Yes, so in plasmid spades, what it does is it does like a first assembly, as for example the normal default of spades, but then it tries to look at the second feature that I talked before. So it tries to look for difference in the coverage of context to classify whether something is plasmid or chromosome. So what it does, it tries to discard contexts that they have a coverage very similar to the coverage that you obtain from the chromosome. And you can only do that because, as Anita said, usually chromosomes are much larger than your plasmids. So if you calculate the median coverage of all your contexts, it's going to resemble to the coverage that you expect from the chromosome. So that actually resembles like a normal distribution. And if you find contexts that are either to the right or to the left of your distribution, maybe you can infer that those contexts, they come from a different replicon unit, so maybe they come from a plasmid. So that's actually the feature that plasmid space is trying to exploit here. So looking at the coverage in order to distinguish plasmids and chromosomes.
So far we mostly talked about classification, so whether a given contact comes from a plasmid or a chromosome, but that doesn't tell you which contacts come from the same plasmid, right? So that doesn't allow you, for example, to assemble complete plasmids or just to classify, to give them identities, right? And why is simple classification not enough? So what extra information can we learn from assigning identities to plasmids?
Yeah, in a classification, we have a binary outcome, either chromosome or plasmids. There can be many different plasmids within a bacterial cell. It depends a lot on the species, how many plasmids we can expect, but we've seen for Enterococcus faecium, at least, we've seen everything from 0 to 11 different plasmids. So if we only have a binary classification, we know this is a sequence from the plasmid or the plasmidome, so all plasmids together, but we cannot really pinpoint from which replicons or from which plasmid a certain sequence or a certain gene comes. So in order to see which one of the plasmids contains which genes, we need to bin the plasmidome even further. And to do that, we developed GPlas, which looks at different features in order to bin these plasmids even further. GPlas takes the assembly graph and then looks at the connections that are there in the assembly graph, but also the K-mer coverage. So how many times a certain sequence was covered in comparison to the chromosome, and also at the K-mer distribution. So the probabilities that were given of a certain contact being a chromosome or being from a plasmid.
So in terms of the information that maybe an epidemiologist might care about, right? Why does it matter, or does it matter whether some genes are on the same plasmid versus on a different plasmid? Like if we try to make this to make use of this information clinically or epidemiologically, does this matter?
Yeah, we assume that, or we know that not all plasmids can be transferred. There are plasmid conjugative plasmids that are transferred from one bacterium to another more easily. So yes, especially if we look at these notorious antibiotic-resistant plasmids, it does matter what they have on there and what this plasmid is.
Mm-hmm. And is the plasmid identity more or less rigid? So if you take a very broad set of isolates of this bacterium, would you observe like a limited set of plasmids, or do they recombine all the time? Do they mix all the time, and do you observe like all sorts of variants of these plasmids?
It does look as if they recombine frequently and exhibit a high level of mosaicism, so a mix and match approach. However, it's also the case that we actually don't have that much data on plasmids yet, because there's a limited amount of bacterial isolates which have been completely sequenced, from which we with certainty know all the plasmid sequences. And a lot of the data is only fragmented because we have those fragmented contigs. So I think there's a lot still to explore about stability or mosaicism of plasmids.
Okay, and so let's talk about GPlas. So this is your tool that you developed to bin these contigs and to assign them to distinct plasmids or replication units. So maybe to talk about how GPLAS works. And maybe the first thing we should mention is that GPLAS works sort of downstream of the tools we already talked about, right? So, for example, ML Plasmids gives you the classification. So it tells you which sequences are from plasmids. And only then does it make sense for to classify these sequences, to classify them further into distinct plasmids, right?
Yeah, that's absolutely right. So in GPLAS, what we tried was to combine different features and different tools that they were already being used in order to predict plasmids. And we wanted to go one step further and to bin those contigs predicted as plasmid into different replicon units. So actually what GPLAS does is first a binary classification, either using ML plasmids or Plasflow. Plasflow is another tool that is also widely used by the plasmid community, in which you can also binary classify a contig as plasmid or chromosome. And that one was also designed for metagenomic samples. So you could also use it for bacterial species in which you don't have a model in ML plasmids.
Right. And that happens even after. So to be clear, the very first stage is assembly, right? So when you get your raw data, actually let's talk in detail about your pipeline. So you get the raw data from the sequencer, right? You maybe do some quality control,
Yes.
trim the adapters and so on. What happens next? So the next step is assembly, right?
Yes, so you will need to do an assembly, for example, using spades or a unicycler, which actually, for example, tries to optimize the spades assembly that you get. And actually, that's the first thing that you need for GPLAS. So there's only one input that you require, and it's actually the assembly graph that you get from for example, spades.
Right, and that's an interesting notion of an assembly graph, and we covered this in some other episodes, but let's sort of remind our listeners and ourselves, what is an assembly graph?
Yeah, so we have been talking about contigs. So contigs are large sequences that come from the assembler, but usually those contigs are also connected, and that connectivity is present in the assembly graph. So in an assembly graph, we have nodes, which are actually my contigs, and those nodes, so those contigs, are connected to other contigs by links, and those links are inferred from the divariant graph, from the assembly, for example, that you can get from spades.
Right, and the meaning of these links is that this contig likely follows that contig in the actual sequence, right?
Yes, that's absolutely right. So in the border of the contigs, what you can find is like there's a reason why the assembler doesn't elongate the contigs that long. So for example, the reason why you cannot get complete genomes if you are using Illumina reads. And that's because when you encounter a repeat sequence, if that repeat sequence cannot be spanned by your read, then the assembler doesn't know whether contig A goes after contig B or contig C, and then it stops there. So that's actually what is delimiting the boundaries of your contigs.
And so you run spades and you run unicycler, and they output this format called GFA. It's a text format popular nowadays, right? So that's just a way to represent the assembly graph.
Yes, that's absolutely right.
And then you run ML plasmids on that graph to tell you, basically to classify the nodes, the vertices of the graph into plasmids versus chromosomes.
Yes, that's actually the only input that GPLAS requires, is like the assembly graph that you get. And after that, it does a pipeline which is fully automated. So it starts with a binary classification, either using ML plasmids or Plasflow. So yes.
Right, and what does it do next?
So then it tries to look for what we called unitigs. So unitigs are particular contigs in which the in degree and out degree of that contig is equal to 1. So what does it mean? It means like there's only one ingoing connection and outgoing connection coming in or coming out from that particular contig. And that's actually very important because those unitigs, so those particular contigs, are we can rely on the coverage that we observe on those contigs. And that's very useful for different features that go after in the pipeline. So that's one of the first steps that we also do. So look into the unitics, so contigs that they have an in degree and out degree of 1. And we can do that because we have the information in the assembly graph.
And coverage here means simply how many times on average this contig was sequenced.
Yes, exactly. Yeah. So what we do is because we also know whether those unitics are from the chromosome or from the plasmid, then we looked at unitics that they come from the chromosome, because in that way we can know what is the difference in the coverage that we expect because of technical problems that, for example, we face in the library preparation or during the sequencing. So those differences in the coverage are not because are due to difference in the copy number, so are due to technical problems.
That's very clever. So you use chromosomes, you rely on the fact that there is just one chromosome per organism, and you use that to calibrate your model or your algorithm.
That's absolutely right. So that's the main concept behind that part of the pipeline. So looking at the difference in the coverage that we expect, not because of difference in the copy number, only because of technical problems. And then what we do is we start walking into the graph. And for doing that, what we do is we consider unitics that have been predicted as plasmid. And then what we do is we start creating walks. So it means if I'm starting for contig A, then I try to elongate these contigs in order to create a path, a walk. And for doing that, what we do is there's usually different connections that you can accept or reject. And those connections are labeled based on the probabilities of a contig of being plasmid or chromosome, but also the probability of that particular contig of being part of that walk. And that's based on the coverage. So we actually assume a normal distribution in which the standard deviation is the difference in the coverage that we observe in our chromosomal contigs. And then what we see is we try to see if that particular coverage of the contig that we want to accept can be accepted or not, depending on the walk that we have been constructing.
So how do you handle the case when the same gene is shared by multiple plasmids, in which case you have sort of the sum of coverages of individual plasmids on a contig?
Yes, that's a very good question. In those cases, what we do is we assign a default probability of that edge. So we kind of say those contigs, because they are shared in multiple sequence, they do not correspond to unitics. So they have an in or out degree which is higher than 1, and then looking at that particular coverage is not useful for the tool. So what we do is we give a default probability to those edges. So in some cases they can be accepted and connected, and later on, if for example you have accepted a connection which is wrong, then the rest of the connections will tell you probably that walk that you have been creating is not the correct one.
So you assign the probabilities to edges, and then you somehow want to disentangle that graph into separate walks with sort of a maximal set of probabilities.
That's actually right. So what we try to see is for each plasmid unitic that we have predicted, we try to create a new set of walks. So we try to see what possible connections do we find in our graph. So for example, in that way, we can differentiate the plasmid part from the chromosomal part, but we can also differentiate, for example, contigs that have a very different copy number rather than others that are also in the same genome, but maybe they have a different copy number. So once we have the walks generated, what we do is we create a new set of links. So we try to see what plasmid unitics are co-occurring in the same walks, and using those links we create a new network. So we try to go from the assembly graph that we have generated, for example using spades, to a new network in which we only have connections corresponding to plasmid unitics.
You also mentioned that you did a benchmark of different tools to predict the plasmid versus chromosome sort of origin of a sequence, right? And I don't know if you also benchmarked if there were tools like GPLAS and whether you benchmarked them, right? And I'm curious, from benchmarking those, did you get any insights that helped you to implement these algorithms, design and implement these algorithms?
So actually we did a benchmarking, but that benchmarking was more focused onto the binary classification of contigs. But in GPLAS, in the manuscript that it's already available in bioRxiv, we did a benchmarking of tools that are that can also be used in order to bin plasmid contigs. So, for example, there are several tools that are out there that can also do a similar job as GPLAS. One is HIASP, the other is MobRecon, Plasmid Spades, or, for example, Recycler. So what we saw is like the combination of looking at the probabilities from the k-mer composition, whether something is plasmid and chromosome, looking at the assembly graph, and looking at the coverage of the contig. Merging those three together is the key in order to do a better binning as, for example, other tools.
How do you combine those sources of data? How do you figure out, when there is a contradiction, for example, between those sources? How, like, in which proportions to combine them?
It was actually quite challenging, especially the first part of looking into the graph. So actually the idea of coming up with walks. So at first, our first idea was to try to create all the walks that they were possible in the assembly graph, but it requires a lot of, it's very expensive from a point of view of computation. So at some point, we also saw like it was more interesting to look into the plasmid unitics and try to look into them. And then if, for example, a contig has been wrongly predicted as chromosome, and it was actually a plasmid, but actually the coverage is telling you that it may actually be part of a plasmid. I think the combination of those two, it's actually what makes GPLAS so successful, because then you are not relying into a single feature in order to predict something as plasmid or chromosome. So you are actually looking also at the coverage and if a contig can be connected or not based on the assembly graph. So I think when we merge those three, what we are actually doing is like if one of the three features is given a contradiction, we still have other two features, and then we rely on the combination of the three to see whether a connection is right or maybe it's wrong.
Are you aware of any limitations with your current algorithm, or is it in a perfect state already?
So what we realized was like there was a main limitation behind GPLAS, and that was when we were exploring plasmids that they have a very similar copy number. So usually plasmids with a similar copy number, they also share repeat sequence. It means like if we look at the assembly graph, those contigs are also going to be connected. And if we look at the probabilities of being plasmid and at the probabilities of being part of the same plasmid based on the coverage, that's actually going to be a higher probability. So at some point in our final network, those contigs are going to be part of the same bin. And that's actually one of the problems that we are still facing with GPLAS.
Yeah, that must be very tough, right? Because do you know any features or any pieces of information that would help you to disentangle this, or is this hopeless?
I think long-read sequencing information could help you disentangle
right.
such plasmids, yeah.
There's actually also something else that we could look at it. So we also talk about the presence of replication genes can also be a feature to predict whether something is plasmid or chromosome. If we will find, for example, the same replication machinery twice in the same bin, that will give you an indication that maybe there are two plasmids merged together.
Right. Oh, that's a very good point. All right, so now you have a tool to predict the origin of a sequence, and you have a tool to bin them into distinct plasmids. So what are you going to work on next?
I think the next step will be to apply it to a large collection of data to determine indeed this transfer of hospital outbreaks, whether we have more plasmid outbreaks or whether we look at clonal outbreaks solely. And I think the bigger advantage of both tools, Plasmid and G-Plas, is that they can be scaled up quite nicely. They don't need to have any manual interference, and we can apply them on thousands of isolates. And also, we're currently working on a new model for Plasmids for Acinetobacter baumannii, which I think will also be really an interesting species.
And what exactly do you mean when you say you're working on a new model? So do you want to just train that on that species, or?
Yes, indeed. So in order to train a new model, we need to have enough complete genomes. And it's either we produce them ourselves or we wait until they turn up in the database. And in this case, we hope to have enough complete genome for Acinetobacter baumannii in order to build a model.
Mm-hmm. And how does the performance compare when you train a model specifically for the species versus you use some kind of generic model, or you use a model trained on a different species? Because you still have those sort of universal features that we talked about, but maybe your machine learning component isn't as great, right? So what kind of performance comparison do you see?
So actually what we have seen this is like, for example, if you use G+ in combination with Plasflow, which is like a general model in order to predict plasmids, so it can also be used for metagenomic samples. In those cases, sometimes you are predicting a contig as part of, as currently predicted as plasmid, so you are actually predicting chromosome contigs as plasmids, and that can really difficult things for G+. So what we have seen is like at some point what you will do is start merging chromosomal contigs with contigs coming from plasmids with a copy number of 1, so copy number similar to the chromosome, and that really makes a difference. So in our benchmarking in the paper, which is available in bioRxiv, we saw like the performance of combining G+ and ML plasmids is much better rather than if you combine G+ together with ML plasmids. So I think the take-home message here was like, the initial prediction that you make is actually really important in order to get the right beans out of it.
Right, but if we talk specifically about the prediction part, so if you take specifically ML plasmids, and if you run a model, if you run the algorithm with the right model trained on these species versus a model trained on a different species, what's sort of the performance difference are we talking about?
Yeah, what we usually see is like, if for example we have trained a model for Enterococcus faecium, for example, that's one of the species that we work in the hospital, and then you try it for a different bacterial species, for example something coming from a gram-negative bacterial species, so a completely different, they are not phylogenetically related. What you see is like the probabilities given by the models, in this case by the support vector machine, are really close to 0.5, meaning like the model is giving you a prediction, of course, it's telling you whether something is plasmid or chromosome, but actually the probability, so the posterior probability of that contig is actually really close to 0.5, which indicates you like you cannot really trust a prediction.
But you have those other sources of information, right? So do they contribute something? Like you should still get higher performance than 0.5 because you're integrating those generic things as well, right?
Yes, exactly. So at some point, if you combine also the probability of the coverage, then it can go higher or lower. So for example, if the assembly graph is quite neat and then you see like that's the only possible connection, then at some point you will accept that connection, so that can also really help. But if you look at the binary classification first, and as you said, what about if you train like a more general model? What we see is like then the problem is much more challenging. But I think it can also be useful, and you can also play with the parameters of G+ in order to relax or being more stringent while you are accepting or rejecting corrections.
Well, Sergio, Anita, that's a very cool work and very important as well, right? So thanks for doing the podcast.
Thank you for having us.
Thank you for the invitation, Roman.